What you are using to watch this video right now is your vision. Vision is what allows people to see the world around them, but what allows all living creatures to have vision? Thanks to multiple components within the eye and the brain, such as the lens, cornea, and optic nerve, your brain is able to process light and turn it into visual images that you can see. But sometimes life is ironic. Sorry. Hopefully this video won't be too cornea. Okay, sorry for the bad puns. That's just how I roll. Anyways, in this presentation, we will go into depth about vision and how it is processed in the body. We hope you enjoy.
So our cell signaling project is about vision, and we hope by the end of our presentation, you'll see us eye to eye.
My name's Gabe.
My name is Dylan.
My name's Evan. This is a basic overview of cell signaling and how it relates to vision. So vision uses the cell signaling process of visual phototransduction, which is an example of signal transduction. So visual pigment molecules are located in photoreceptor cells, which are rod cells and cone cells. And these will absorb photons which get converted into electrical signals. Both cone cells and rod cells are located in the retina of the eye, and their shape has long and narrow tubes that have an inner and outer region. The inner regions of these cells contain the nucleus and lots of mitochondria, which contain ATP, as ATP are required for this process. In addition, the outer region contains a membrane with rhodopsin and a ligand-gated ion channel in order to lower the membrane potential generated from the inner regions of the cell.
So this is the cell signaling pathway. Okay, so reception to response. To start off, photons are the ligands for the cell signaling pathway in the eyes. A molecule present in the rod cells, rhodopsin, is the G protein coupled receptor. To preface this, and I'll get more into this later, but eyes can detect between light and dark through neurotransmitters, which is regulated by cyclic GMP and calcium channels, which are opening and closing. So when the photons of light hit the rod cells, the photons strike the membranes of the rod cells, which are lined with rhodopsin, and this can excite the G protein, opspin. And cisretinal means inactive, and transretinal means active. And you can see this on the image above, which is a chemical structure. So opspin basically activates the enzyme phosphodiesterase, which converts cyclic GMP to GMP. This means that the calcium channels cannot be opened, which means that no neurotransmitters are saying it's dark to the brain. So finally, synaptic responses of the light being present are sent out to the brain with the synaptic center, which is on the image on the very right at the very bottom. So when photons of light aren't detected, cyclic GMP is integrated because there's no enzyme, and this opens the calcium channels, which lets the calcium 2 ions flow in, and the neurotransmitters saying, oh, it's dark, there's no photons, which is exited out of the cell. So this is just a summary of what I said. There are two options which can basically happen with photons being present and not being present.
So there are a few faulty mechanisms with the eye. First, the body's inability to blink due to the action of blinking eyes or produce aspirin. Aspirin and blinking eyes are very important for the eyes as they keep the eyes moisturized and keep constant signaling of rhodopsin. And if not, photons continually come in, causing rhodopsin to become saturated. And the body has a natural response to this, which is to create tears, to add moisture to the eyes and produce excess rhodopsin, which can cause blurred vision from the tears. And to fix this, a darkened light cycle or opening and closing the eyes quite often can help prevent this saturation. Another faulty mechanism is the interruption and or damage to certain aspects along the cellular pathway that can hinder your eyesight. So the cause of this is abnormal optic nerves, and the symptoms are light sensitivity or photophobia, reduced contrast vision and acuity, visual field loss, and reduced depth perception.
So some more faulty mechanisms are these factors that affect vision, including aging, damage, which is such as like an injury where you damage the retina or clouding of the cornea or lens, developmental disorders, disease, infection, and refractive errors. And basically all of these will lead to impairment of the vision, and it'll impair one's ability to see because it can interfere with the light entering through the eye or the ability of the eye to focus. So another faulty mechanism is visual field loss, and this is when part of the visual field is missing, and the visual field is the area when one's eyes are fixed in a position so they're looking straight ahead. And this basically happens when there's interruptions inside of the brain, such as when there's damage to the right side of the primary visual cortex, which results in the field loss on the left side of each eye. And on the right are 3 terms to describe different types of visual field loss. This graphic right here shows examples of visual field loss, and as you can see, they're labeled with the different parts of the brain that they're affected by. And now moving on to different graphics of the pathway of vision. On the top left corner, we can see an image of normal vision and the pathway it takes through the eye. First, the light enters through the cornea, then from the cornea it passes through the pupil. Then from the pupil, the light passes through and hits the lens, which is a clear structure inside the eye, which focuses light rays onto the retina. From the retina, the light passes through the vitreous humor, and from the vitreous humor, the light reaches the retina, which is a nerve layer that is sensitive to light and lines the back of the eye. The signals received are then carried by the optic nerve to the visual cortex of the brain. And in this image in the bottom right, we can see the lateral geniculate nucleus, which is located in the thalamus of the brain. And this is where all the sensory information stops to enter, and it's where the visual signals are organized and sent to the primary cortex, or the visual cortex. In the visual cortex, there are 6 layers which allow for depth, form, color, and motion to be perceived. And this is where the signals received from the retina are processed, and it leads to interpretation of visual information. And on the right here is a diagram that Gabe drew, which talks more about the reception to reaction of the rod cells that Anirudh talked about earlier.
So next we're going to be going over the current direction of research. So the overview of scientists are researching in terms of vision and eyes in general are the cornea and how the cornea interacts with specific proteins, gap junctions, which are basically the interactions between different types of animal cells, and cell signaling. So cell signaling in vision. What scientists have been researching is the relationship between color and photoreceptor cells. Somewhat recently, there's the discovery that rod cells actually have a unique pigment that's sensitive to green light, which nobody expected before. Additionally, there's ongoing research about retinal arginase signaling, which protects the nerves and blood vessels in the eye in the event of an injury to the retina. These are our references.
Thank you for listening.